So the nervous system has two general divisions, or the peripheral nervous system has two general divisions, the autonomic and the somatic. All right, so this will be the autonomic, and here's our somatic. Now the autonomic is also called what? How do we describe the autonomic? Is it voluntary or is it involuntary? It's involuntary. Perfect. It's involuntary. It's also called the involuntary part of the nervous system, whereas the somatic is called the voluntary. And that simply means that we have control over the somatic system and we do not over the autonomic. Now, when you look at the two types of neurons in the autonomic nervous system, what are the two subdivisions there? There are two subdivisions of the autonomic nervous system. Parasympathetic and sympathetic, right? So parasympathetic, and we got sympathetic. Now, the fibers of these two neurons arise from what part of the spinal cord? What part of the spinal cord do these fibers arise from? The lateral horn. Perfect. So the lateral horn, the lateral horn. And how do they enter the spinal nerve? How do they enter the spinal nerve? Through the dorsal root or the ventral root? It was through the ventral root, right? So we'll put ventral root, so we know that it's coming out through the front of the spinal cord and getting into that spinal nerve, right? So the ventral root here. Now, there were two different types, two different lengths of neurons. The sympathetic system had a shorter preganglionic neuron and a longer postganglionic neuron, whereas the parasympathetic system had a longer preganglionic neuron and a shorter postganglionic neuron. Okay, the somatic nervous system, the fibers arise from the ventral horn, not the lateral horn. And then there's just one neuron. This is called a motor neuron. Right? There's no pre, there's no post, one neuron, the motor neuron. Now, when we speak about the different effector tissues, what's the effector tissue of the autonomic nervous system? What type of tissue does the autonomic nervous system innervate? Smooth muscle, what else? Cardiac muscle, glands, right? So smooth muscle, cardiac muscle, glands. And then specifically the sympathetic also innervates adipose, right? Adipose here. Now let's talk about the sympathetic nervous system. What do we call this region here, where the preganglionic neuron synapses onto the postganglionic neuron? We call this an autonomic ganglia, right? This is an autonomic ganglia. And a collection of these in the sympathetic system is called what? The collection of the autonomic ganglia of the sympathetic system, what do we call that? That collection? The sympathetic chain, correct, the sympathetic chain. And now we spoke about three routes of communication for this preganglionic neuron to get to its effector tissue. So we spoke about three things that can happen with this preganglionic neuron. What were the three things? So one is going to be the sympathetic chain. Right, it's going to synapse onto the sympathetic chain. What were the other two routes or the other two alternative options? So one was the adrenal medulla, the chromaffin cells, and the collateral ganglia. Perfect. And the collateral ganglia. Okay. Now, we also spoke about the type of receptors, so the neurotransmitters and the receptors involved. Now, what type of neurotransmitter is released from the preganglionic neuron? Acetylcholine. So this is what we know, as fact. All preganglionic neurons and all motor neurons secrete acetylcholine. So we can go ahead and put acetylcholine as the neurotransmitter on all of these neurons. All preganglionic neurons and all motor neurons. Okay. What about the postganglionic neuron? What about the postganglionic neuron? What does it release on the sympathetic side, let's say? Norepinephrine. Good. So on the sympathetic side, norepinephrine is being released at this junction. What about on the parasympathetic side? Acetylcholine again. Perfect. Acetylcholine again. And then the somatic system only has one synapse, and that's always acetylcholine. Okay. Now, what type of receptor is found on the second neuron here? Nicotinic cholinergic, right? Nicotinic, and then this is always nicotinic. What type of receptor is found at the neuroeffector junction, so between the second neuron and the effector tissue? What type of receptor does this norepinephrine bind to? Come on. Adrenergic, good. So adrenergic are on the sympathetic side. And what about the parasympathetic side? We know it's a cholinergic. Muscarinic, yep, muscarinic. Right, so we have nicotinic receptors on all of our postganglionic neurons and on skeletal muscle. And then on our effector tissue on the sympathetic side, we have adrenergic receptors. And again, we have different flavors of those adrenergic receptors. And then on the postganglionic parasympathetic side, we have muscarinic receptors. Right. Now let's talk about the differences here lastly. So let's talk about the difference between the neuromuscular junction. So this is the neuromuscular junction. Right. Here would be the neuroeffector junction, right? Both of these are called the neuroeffector junction. And what were the differences? We mentioned two differences between this neuron to neuron synapse and then this neuroeffector junction. So what were the two differences we mentioned between these two synapses? Two specific differences. So the neuromuscular has the end plate, yeah, but what about the neuroeffector junction was different than just a neuron to neuron synapse? It's a greater distance, right? So greater distance. And something else. There was the presence of a structure that's not seen at a neuron to neuron synapse. What do we call those structures? Varicosities, right? The presence of varicosities. Okay, so these are the two main differences between a neuroeffector junction and a neuron to neuron synapse. Alrighty. And let's put our effector tissue. So our effector tissue for our smooth muscle, or for the somatic nervous system, we know is going to be skeletal muscle always. Okay. All right, any questions here? Any questions on this? Oh, two more things we forgot to touch on. So what part of the spinal cord is responsible for sympathetic output? What part of the spinal cord? Is it craniosacral? Is it thoracolumbar? So this is going to be T1 to L2, right? Also called thoracolumbar. And then this is going to be cranial nerves 3, 7, 9, 10, and S2, S3, S4. And this is hence called the craniosacral outflow. Okay. And then obviously we should know the functions of both of these divisions. Sympathetic is going to be fight or flight or freeze. Parasympathetic is going to be rest and digest. And we know that the somatic nervous system is our voluntary skeletal muscle. Okay. All right, so the next thing we'll talk about, any questions on this? Sure. Any additional questions on that? Now you want to make sure you can differentiate the differences between the steps at this junction and the steps at this junction. Okay, specifically what receptors are involved and what neurotransmitters are involved. All right. Okay, and the last thing we're going to touch on is these receptors, the adrenergic receptors, and how we classify those and their function. Are we good with this picture? Question? Here? Oh, muscarinic receptors versus adrenergic receptors. Okay. Okay, so the next thing we'll talk about are those adrenergic receptors. Now, adrenergic receptors are what two general types? What are the two types of adrenergic? What are the two general types of adrenergic receptors? Alrighty, so we have alpha receptors and beta receptors, specifically alpha 1, alpha 2, beta 1, beta 2, and beta 3. Okay. What are the locations of these receptors? What are the locations here? So we know alpha 1 receptors are the most numerous of all receptors. And they're found pretty much on vascular smooth muscle. Okay. Alpha 2 receptors, same deal. Most numerous, so alpha receptors in general, the most numerous, and they're all found on vascular smooth muscle. Okay. What about beta 1 receptors? What organ? Heart and kidneys. Beta 2 receptors. Think about your breathing. Lungs, and then also your uterus. Okay. What about beta 3 receptors? This is adipose tissue. Okay. Let's talk about their affinities. So we know they have differing affinities for norepinephrine versus epinephrine. So alpha-1 receptors and alpha-2 receptors prefer what neurotransmitter? Norepinephrine. Both of these prefer norepinephrine over epinephrine. Okay. What about beta-1 receptors? Equal affinity. Good. So beta-1 and beta-3 have equal affinities for norepinephrine and epinephrine. Beta-1 and beta-3. And then beta-2 is sort of the unicorn. What is its preferred neurotransmitter? Epinephrine. So beta-2 prefers epinephrine over norepinephrine, right? Let's talk about their second messenger. So this is their signal transduction pathway. What were the second messengers involved in alpha-1 receptors? Remember, alpha-1 had an entirely different mechanism, an entirely different set of second messenger. This was IP3 and DAG, right? IP3, and then of course DAG. And then this was a G stimulatory protein. What was its effect on the tissue? Excitatory. Okay. What about alpha-2? Alpha-2 had an entirely different second messenger, didn't it? What was the other second messenger we talked about? So this is going to increase its second messenger. Let's make a note of that. What was the other second messenger we talked about? So we only spoke about two. We spoke about IP3. We spoke about cyclic AMP. So cyclic AMP is involved in all the other receptors. Right? Whether it goes up or down is going to differ, but it's involved in all other 4 receptors. Okay. In alpha-2, it's going to be decreased. It's associated with a G inhibitory protein. And so alpha-2 is going to decrease cyclic AMP's second messenger. What type of effect is that going to have on the cell, though? Although it's decreasing its second messenger, what is its effect on the tissue? Not inhibitory. Alpha-2 is like that exception to the rule, right? Decreasing its second messenger is excitatory to the tissue. Okay, when you think about a blood vessel, we're going to talk about smooth muscle in the next block, but when you think about a blood vessel, the smooth muscle in that blood vessel has a natural tone. So naturally it wants to constrict, okay? And so inhibiting the second messenger on these vascular smooth muscles is going to open up the blood vessel, which is an excitatory action. So although its second messenger pathway, its mechanism of action is decreasing cyclic AMP, it's still an excitatory effect on the tissue. Opening up of the blood vessels is excitatory, okay? Let's look at beta-1 and beta-3 together. So they're both going to increase their second messenger, right? G stimulatory proteins, and that's going to have an excitatory effect on the tissue as well. Beta-2 is that second sort of unicorn. Although it is increasing its cyclic AMP messenger, what is the effect on the tissue? Inhibitory. Inhibitory. And the example I like to give is in the lungs. When we increase the cyclic AMP of the smooth muscle in the respiratory airway, that actually causes that smooth muscle to constrict on itself, right? And in constricting on itself, it's closing off the airway. That is an inhibitory effect on the tissue. So the contraction of that muscle brings about an inhibitory effect on that location, on that effector tissue, okay? So don't confuse the second messenger effect with the effect on the tissue, the effector tissue. All right, any other questions? Any questions on this bit, on the adrenergic receptors? Which one? Top. This one? This is the same. Most numerous and also found on vascular smooth muscle. Now we want to bear in mind here, just as a side note, that these are all muscarinic receptors, meaning that the receptors are not a channel, but instead the receptors are coupled to a G protein, right? So there's that sequence of events that takes place inside the cell after norepinephrine binds to these receptors. All right, any additional questions? I think that's all I'm going to review. As far as the skin, tissues, and tegumentary system, that was pretty fact heavy. So just go over that, make sure you know the layers, make sure you know what's going on in each layer and the function. Any other questions, any of the topics that were unclear?
Hey, can you go over that?
Trap. All right, so when we were talking about TRAP is one of the mechanisms by where we can increase force by increasing the frequency of stimulation. And all that means is that the faster we send action potentials, the faster we stimulate this muscle, the more force we generate each time successively, right? Now, TREP is unique. It's different from summation in that when we stimulate this muscle the first time, we generate X amount of force, okay? The muscle completely relaxes, meaning that all the calcium has left the cytoplasm and we go back to rest. Now, we come along, stimulate this muscle a second time. For some reason, we generate a little bit more force this time. Although the muscle completely relaxed, the next time we stimulate it, we generate even more force. And that continues to happen until we plateau at the maximum force that is allowed for this muscle. So this is a mechanism by which stimulating a muscle repeatedly, more frequently, increases the tension that's generated, increases the force that's generated until we plateau at the maximum force for that muscle. Okay, it's important to distinguish TREP from summation. If you look at summation, this tension does not return to zero, meaning that all of the calcium did not fully leave the cytoplasm. At this level, when we stimulate it again, when we stimulate that muscle the second time, there's already some calcium in the cytoplasm from that first stimulation, from that first action potential or end plate potential. And so the next time we stimulate it, we ride on the previous tension. So we just increase from the previous tension and successively generate more force each time until we completely max out this muscle, which is called complete tetanus or fused tetanus. All right. Does that answer your question, Ryan? So the key point with TREP is that in between muscle contraction, we have full relaxation, but the next time we stimulate that muscle, for some reason, TREP is not fully understood. For some reason, we generate more force. Okay, we can understand summation. Summation makes sense. If there's still more calcium in the cytoplasm, it only makes sense that the next time we send an action potential, we generate more force. But for TREP, where there's full relaxation in between, why the next time we generate more force is not quite understood. We just know that this happens.